So to pick up from where we left off last time, we're talking about experimental designs. Remember, experimental designs are when we're looking at cause and effect relationships. So we're looking at, hopefully, the cause being our independent variable and the effect being our dependent variable. Specifically, we're looking at between subjects designs. Between subjects designs are those kind of simplistic, the very typical ones that we think about when we're looking at two separate groups of people. And we'll have maybe a control condition and an experimental condition, a control condition and a treatment condition. And again, the most basic form of this is two groups, but we can have two or more. So in between subjects designs, we are, again, and these are also synonymous with independent groups because, again, we're looking between independent groups to see if there are any differences. The advantages to these, again, that there's no concerns about practice, experience, fatigue, etc. Because, again, we have separate groups participating. So we don't have any risk of them getting bored with it because they're getting it over and over again, learning about it, any of these kinds of things. Again, these are really, really simple. We're not dealing with really complex designs. We're not dealing with a lot of complexities here. We're just dealing with at least two groups, but sometimes more. The disadvantages to these, and our major disadvantage, and the one that, again, I want you to most remember when you're thinking about this, is the difference between the individual subjects or those individual differences. Again, we try to best control this through randomization and random assignment to our conditions as the way that we're trying to control for that. But that's the thing that we always have to be most concerned about is our groups, were they different to begin with? Did our levels of the independent variable have no effect at all because they were different at the start? And again, we're using random assignment to really kind of control for that. The other disadvantage, again, is there's more participants needed. If I have two conditions and I need 20 participants in each condition, then I need 40 participants. If I have 5 conditions and I need 20 participants in each condition, then we're at 100. You can see how this adds up really, really quickly and can be very additive and create a large, large number. So when we're looking at these individual differences and we're looking at variability between populations, this can cause kinds of issues. So your book talks about how a 10-point difference between two different groups can be very different depending on the population that it's drawn from. So these are figures from your books or tables from your books. On table 8A, and again, population A is the individual differences are relatively small. So we have a group here that has very low variability. Remember, variability from 3510, we're looking at how much the group as a whole varies. Whereas in population B, if you look at the scores, there's got relatively large difference. Population A, it looks like we're varying from maybe 38 to kind of the highest number I'm seeing is maybe 42. So we're really pretty tight there. Whereas in population B, I see numbers as low as 24 and I see numbers as high as 56. And there could be more variability than that. So we're dealing with two different kinds of populations. When we're then selecting our samples from our populations, we're going to select 20 random people from population A. We'll divide them randomly between the treatment group and the condition group. We'll have 10 people in each. And then we'll do the same subsequently for population B. If we did this from population A, again, our groups that have very low variability, if we have a treatment effect of our study, whatever our independent variable is, it creates, we have a score of 10 higher for the people who are in the treatment condition. So that because our population has relatively low variability and we have low differences, we have low individual differences, we can see that 10-point difference very, very broadly. It really divides those groups very nicely, and you can see your treatment effect very, very clearly. However, if we pull 20 people from population B, again, that has lots of variability, and we randomly assign 10 to the treatment condition and 10 to the control condition, and again, we can even have that same 10-point treatment effect. And so we add 10 points to kind of everybody that we selected from the control condition, just as, again, an example. And we no longer see that nice division of control and treatment group because this population has so much variability. Our 10-point treatment effect is really lost, and we don't see that. And so again, we're going to have differences, and this is where individual differences in the population can really cause problems in our studies. And this, again, one of the biggest disadvantages we have to this, because if you're drawing from a population that is very, very variable, and you've got a relatively small effect, you can lose that treatment effect in this very variable population. Whereas if you're drawing from a population that has very low variability, again, you can see those treatment effects a little bit easier. So a lot of this depends on, and deciding between a between subjects and a within subject is, and to some extent, is going to depend on what kind of population you're dealing with and what kind of questions you're dealing with. So again, the issue here becomes that the differences between the conditions and the variances within the conditions. So we want the differences between our conditions, between our control and our treatment conditions. Hopefully we want those to be kind of high because that's where we've done our manipulation. That's the place that we've created, hopefully, a difference between these groups. Where we want our variances within our conditions relatively low, hopefully, because it's kind of our noise. So when we get to the statistics point, we're going to kind of, part of the course, we're going to kind of talk about signal to noise ratios. And if you're not familiar with that term, signal is kind of what you're looking at. That's really what you're trying to see, the signal in and amongst the noise. So in this example here, we're talking about the differences between conditions. That's our signal. That's what we're looking for. We're looking for that signal. And the variances within conditions then is our noise. So we're trying to see this signal in and amongst this noise. If you have a small signal and a lot of noise, it's really hard to see. If you have a really strong signal and a lot of noise, you can see it. And if you have a really strong signal and a little bit of noise, you can see it. But you can also see a weak signal in a little bit of noise. So when you have a small treatment effect and very little variability, you can really see this. And again, our signal is we're looking at these between conditions, the effect of our independent variable on our dependent variable. Our noise really is these individual differences. The difference between Bob and Sally and Joe and Tim, those are individual differences. And do we have the same makeup of those in our treatment condition versus our control condition? Is our population very variable to begin with? Again, do we have a large amount of variability? One of the reasons that the infant research that I've done, that we typically try and do those as within subjects or repeated measures designs, which is our next chapter, where again, everybody's getting both the control and the treatment condition, is because babies are really very variable. They have a huge difference in looking time. Some kids look, 10 seconds and they got something. Some kids look 60 seconds. And that's a big difference. And so if we have a kid that's looking 10 seconds and they have an increase of looking, well, their increase might be a couple of seconds. Where if we have a kid that's looking 60 seconds and has an increase of looking, a couple of seconds isn't going to mean much for that kid. So again, that's a lot of individual differences in that behavior. And because we have a lot of individual differences, a lot of noise in that behavior, we really try and look at this in a different way. We try not to look at them in terms of between subjects designs because it is very, very noisy and we can't really see these signals as well. So we try and do these within subjects designs because then we can minimize that noise and really look for that signal. Another way that we can kind of deal with this is trying to minimize our variance within conditions. Again, a lot of this is coming back to selecting participants equally and treating them equally. So by standardizing procedures and treatment settings, we're treating everybody equally. We're limiting the kinds of other variants that could create differences within our conditions. We really just want our variants to be just those individual differences and really minimize any kinds of differences between our groups. We can also kind of try and limit individual differences. We can kind of select samples or from populations that are a little bit more limited to begin with. Sometimes that's why college students can also be a really great population to deal with, because we have a range of kids here, of students, that's a lot more narrow than if you look at the entire population in terms of education, in terms of IQ. We have a more limited, a less variable range in a college setting than we do in the whole population. Again, random assignment and matching are going to be really important. And it depends, again, which one you're doing depends on the kinds of research question that you're asking. But it's allowing us to really minimize this variance within our conditions, because everybody had an equal shot of being in the two conditions. Or we've paired you off because of some particular matching trait. And again, this allows us to kind of equalize those individual differences between those two populations, so that hopefully any differences that we see are really just due to the effect of our manipulation of the independent variable. Sample size will also be an issue that can really affect these things. And that's because the bigger your sample is, the closer it approaches to infinity. Well, obviously you don't have a sample that's approaching infinity, but the bigger that population gets, the more normal it becomes. And when I say normal, I mean like the normal curve. So you're all familiar or can think back to 3510 and when you learned about the normal curve, and in some of your other math classes as well. Again, that's that bell curve. And the more participants you have, the closer you get towards infinity and the closer you hit to that normal curve. So this really is an issue of probability. And the closer you get to that, the more normal these things become, and the easier it is to really control for these individual differences, because you'll just have a few people that are in the extremes and most everybody will be in the middle. Whereas if you have a really small sample, you may have accidentally got, one of these extreme cases, somebody that's in the tails of your normal curve. And that's one of your 10 participants. So a tenth of your group is an extreme. So again, that's not indicative of a normal curve. So again, the more participants you have, the more normal it becomes, and it really does help reduce some of these variability issues. So let's move on to threats to internal validity. Again, I asked you to review these online, the online tutorial, and I hope you've done that. But let's review this some as well. So some of the issues that we have, again, threats to internal validity, is when we've done something and it has jeopardized our ability to make these causal inferences. Remember, the whole reason we're doing this experiment is because we're wanting to say that our independent variable, what we're manipulating, is causing a differential effect on our dependent variable, what we're measuring. Again, we're trying to say that A is causing B. And to do this, we really need to, again, control these threats for internal validity. So when we don't, we lose the ability to make these causal inferences. One of the main ways we can do this, and we talked about this already with natural groups designs, is when we're dealing with intact groups. When we're dealing with a group that's already there, if we're dealing with second graders versus fourth graders, there's a whole bunch of different things that are going on with second graders and fourth graders. Those are intact groups. When we're dealing with psych majors and chemistry majors, well, those of you who are psych majors picked psych because there's something in psych that really interests you. There's something that in psych that's going to be helpful for you. There's a reason that you're in psych, and there's a reason chemistry majors are in chemistry. And those are different reasons. You aren't in psych because you really love chemistry and you're really into chemistry, and you're not in chemistry because you really want to be a research psychologist. Again, these groups are different, and these issues and these different variables with these intact groups can vary then with our manipulation of our independent variable. And so we lose the ability to make these causal inferences. Again, anytime we don't control for any of these extraneous variables, they can become confounding variables. So again, anything that we're allowing to co-vary with our independent variable or our dependent variable can become then a confound. Again, if we're really controlling for these things, if we're balancing it out, if we're treating groups equally, then all those extraneous variables kind of fall out the bottom because everybody has them. We see them in both the control condition and the treatment condition. And so because they're both there, they're both noise, and they cancel each other out. Whereas if they're differential between the two groups, then they can come across as signal and instead of noise, and that can be a problem. And we lose again the ability to say that A is causing B. Another issue is when people drop out of your study. Attrition is something that you're going to have, especially if you're doing a study over long periods of time. Even if you're doing someone over short periods of time. The computer is not going to work sometimes, and you're going to lose that subject. Sometimes, something weird can happen with the power. Maybe you had people scheduled, last Wednesday to participate in your study, and there was no school last Wednesday. And then you had other people scheduled last Thursday, and there was no school last Thursday. So again, because the university closing, the weather caused issues. And if you had only been doing your treatment conditions on those days, and you have your control conditions done another time, we've created a selective loss between these two situations. And our groups are no longer equal. Our groups are no longer the same. They've had different situations. And because this selective loss occurs, can cause issues. We'll talk about this one in a little bit more detail on some of the subsequent slides. We also have issues with demand characteristics and experimenter effects are not controlled. Again, these are things that we'll go into and talk to more detail about. Experimenter effects are going to be things when the experimenter has an idea of how they think the outcome is going to be, and that's not controlled for. And so they're responding differently to the treatment condition versus the experimental condition. Demand characteristics can be any number of things that, again, we'll go into in a little bit more detail. So again, with intact groups, these are the groups that exist before the experiment. These are those natural groups designs that we talked about in the previous lecture. Examples of these include maybe kids from different classrooms, departments within an organization. So maybe we're looking at just, within the college, the psych department versus another department. Again, these are intact groups. We can't compare those because there's differences between those groups inherently. And those differences can be then measured maybe in our dependent variable. And so we don't know that it's our independent variable that's caused those differences. Sections of various courses, any of those things are examples of intact groups. We lose our ability to make these cause and effect inferences because these intact groups do not allow for random assignment. This can also be things like the other natural groups, male, female. We talked about schizophrenia versus non-schizophrenia. Any of those kinds of things. Those are also intact groups as well. But again, because in both situations we lose the ability to randomly assign, we can't say that these groups were essentially equal before we started. Because we don't know, we're not able to say that, we're not able to control for these individual differences, we can't make these cause and effect judgments. And internal validity has then been diminished greatly. When we've got these intact groups, we don't have this random assignment. Again, these subject characteristics are not balanced. If we look at psych majors versus chemistry majors, my guess is, and I don't know this, my guess is that there's going to be a proportional difference in gender between those two groups. That there's going to be more females to males in psychology, and there's going to be more males to females in chemistry, would be my guess. Those are subject characteristics that are not balanced between these two groups. Again, because we've lost these intact group, we've lost this ability for random assignment, we're no longer able to make cause and effect judgments. If you are trying to make a judgment about cause and effect, if you want this to be an experimental design, do not use intact groups. Intact groups create a situation where you're no longer experimental. We're into quasi-experimental. We might even be pushing that. But again, do not use intact groups if you are interested in this, in your research being an experimental design. Extraneous variables, again, are every other variable that you're not either manipulating or measuring. Now, they're always there, no matter what you do, no matter how well you control for them. Those extraneous variables are always there. But again, you're trying to control them. You're trying to equally distribute them between the two groups so that they're not causing any differences between your two groups, and that we're really minimizing that noise, so that you can see that signal of the differences between your groups. And these can be anything, any practical considerations that you have to think about. Which rooms you're in, who your experimenters are, any of these kinds of things. These extraneous variables are back to the last lecture where we talked about treating participants equally. We treat participants equally for a lot of reasons to control these extraneous variables and to keep them from becoming confounds. Some examples of these include the number of participants in each session. So if you have, 3 participants when you have your control condition and you have 25 participants when you do your experimental condition, that's very different. They're getting very different situations. You really need, those are kinds of situations that you need to control for and really kind of equate for that. Having different experimenters. If you have experimenter A does the control condition and experimenter B does the treatment condition, well, maybe it's not that your control condition and your treatment condition are different. Maybe it's that your experimenters are different. So again, those are things that are extraneous variables and that you need to control for in some way so they don't become confounding variables. In the case of the experimenter, you could have experimenter A do half the subjects for the control condition and half the subjects for the treatment condition. And experimenter B can do half the subjects for the treatment condition and half the subjects for the control condition. So any differences that we have between experimenter A and B are balanced equally across the two conditions. And then again, this noise will cancel each other out. Different rooms where they're conducted. Again, this could be any of these practical considerations. The list for this can go on and on and on. And it's going to be very, very specific to the kind of research that you're really doing and the kind of research question that you're actually asking. So back to our example of the 2 experimenters. Suppose we have the 2 different experimenters, and again, A does all the participants in the treatment condition, and B does all the participants in the control condition. What we then have is a situation where the experiment has become confounded. That extraneous variable of experimenter has now been allowed to co-vary with the levels of our independent variable. By assigning one experimenter to the treatment condition and one experimenter to the control condition, we've allowed a third variable, in this case experimenter, to co-vary with our manipulation of the independent variable. Because of that, we know that this experiment is confounded. Any differences between the two on the dependent variable may be due to our treatment condition. It may be due to our manipulation of the IV, but it might also be due to our experimenter. So again, by allowing these two things to co-vary, we've really created a problem. Again, the way around this would be we would have experimenter A do half of each condition and experimenter B do the other half of each condition, because then again, we've balanced that experimenter variable equally across the two. So again, we've balanced that, and then that noise cancels each other out, so we can really still see any signal that's there. Subject attrition, again, this is when people drop out of your study for any possible reason. Maybe your treatment condition is really boring and your control condition is not. And so people drop out of your treatment condition because it's boring. But people don't drop out of your control condition because it's fine. Again, that would be attrition. And as long as you have people dropping out of your study, and you have this loss is equal. So again, things like computer problems. The computer's not going to really just affect the treatment condition or the control condition. They're going to affect both. It's going to be equal loss. We're going to have participants lost from both conditions equally. And as long as that's happening, it's okay, because again, these groups are being treated equally. The problem arises is if whatever group equivalence that we had at the start of the study is destroyed. So again, if we have selective attrition, if we have people dropping out of our treatment condition and not our control condition, that's a problem. If we have people dropping out of condition A, but not condition B and C, that's a problem. Because that random assignment that we did at the beginning has basically been shattered because the people that dropped out of the conditions, they're dropping out for a reason, and they're dropping out differentially, and something about them has created a situation now where our condition A is not equal then to condition B and C. Our groups are no longer the same because we've lost some of this. Differential attrition is one of the things that we call this, or selective attrition. Again, this is where we have different attrition rates for different groups. So again, more people are dropping out of your treatment condition than your control condition. More people are dropping out of condition A than conditions B and C, those kinds of things. Because our groups are no longer have that same equivalence that we started out with, this becomes a problem. We could see this being an issue. The differential attrition can also be an issue within subjects' designs as well. And we especially see it in longitudinal designs. Again, longitudinal designs is a really kind of like super version of a within subjects design that can happen over many, many, many years. And so again, attrition is going to happen in those kinds of studies. The problem is again, when it's different, when that loss is not equal. So when that loss is not equal, we lose internal validity. And so again, we lose the ability to say that our independent variable is causing our dependent variable. Again, this occurs when they're lost differently across conditions. There may be some characteristic of the participant that's responsible for the loss. Again, maybe the participants that have really demanding schedules are the ones that are kind of dropping out because they are really have a lot of stuff to do and they don't have time to do this really annoying study too. And so they're dropping out. That's really would be nice to have those people in there because they have different characteristics. And again, if they're dropping out differentially between these two groups, if we're losing the demanding schedule people in treatment group, but not in our control group, our groups are no longer equal. This can especially be a problem if within our subject characteristic that we're looking at is related to our dependent variable. So if we're looking at something like maybe responsibility at work or some sort of parental feature. Again, if we're dealing with people that have really demanding schedules, they may have really demanding schedules because they have a lot going on at work. And so if we're looking at some measure of employment, that's a problem because having a lot going on at work and looking at employment are going to coincide. We're going to have these relationships between these. So this is especially problematic in these situations. And again, these create larger threats to our internal validity. Another example would be suppose a treatment for depression is compared to a non-treatment control condition. Selective subject loss might occur if people drop out of the control condition more than the treatment condition. That would be a problem. Again, we've created a situation where these groups are no longer equal. Mechanical subject loss is what I was talking about with the computers. When some equipment failure or some experiment or error creates a situation where we can't get the data. For whatever reason, the computer won't work, the power's not working, an experimenter's late, experimenter codes it wrong, any number of things. These are often due to chance factors. These aren't something that are going to be varying with our independent variable or our dependent variable. And because of this, they typically occur pretty equally across conditions of the experiment. We're not going to see one condition having a lot more loss than another condition, if we've really done a good job of kind of balancing out our conditions. Now, if we're doing, all of our treatment condition first, followed by all of our control condition, well, then yeah, we may have a problem. But again, that's another reason that we're going to kind of create random orders for conditions as well, so that things like mechanical subject loss are going to be more equally balanced over. There may be times where the computer is just not working. Every time something's wrong, it's a bad week. And so again, if you have both your treatment and control conditions collecting data for both of them that week, then we're going to have equal loss across those two groups. So if you've done a good job of creating this randomization for assignment to your conditions, that will be beneficial. Because this loss is due to these chance events, it does not typically threaten the internal validity of the experiment, as long as it's not allowed to co-vary with your independent variable or your dependent variable. It's not a threat because it is due to chance, and it's typically equally across the two groups. Again, if you had a situation where you lost all your treatment condition and only part of your control condition, that would create unequal groups, and then we would have an issue at hand. But again, typically this is not really an issue that's going to really affect this, as long as it's handled appropriately. Experiments or effects. There's potential biases that occur when the experimenter knows the hypothesis, when the experimenter did the hypothesizing, and what we expect to see. Well, if you're doing a study and this is your study, this is your brainchild, this is your idea, this is what you think is going to happen. Well, you've done a lot of work on this study and you really want to find something significant. And so you may not mean to, but you may inadvertently kind of create a situation where you're behaving differently to the participants, or you're coding things differently. And again, this may not be on purpose. This may be accidental, but it creates a situation where these experimenter can create unequal treatment of the two groups. And again, when we have unequal treatment of the two groups, then we no longer have this equal treatment that we're trying to do. We're creating more noise, and so then it's going to be a lot harder to see that signal. One of the ways we can handle this is to keep our experimenters blind or unaware of the expected results. So when I was an undergrad, I was working at a lab, and the people that we had actually coding, the researcher had coding the actual infant behaviors, were the very first people, the newest people to the lab, the people that had been there the shortest amount of time. We trained them on what a look was, what a look wasn't, and we just had them record looks and no looks. We didn't want them to know anything else about the research. So they were really blind to the kinds of research what we were really looking at, all of these kinds of things. We just wanted to know if they knew if this baby was looking at this or if this baby wasn't. When I was in graduate school, because the coding was a little bit more complicated, we had either the graduate students or the really people, the undergraduates that had been there a really long time, were the ones that were coding the behaviors. In that situation, we were just blind. I mean, I never assigned a kid to a study that I was running, so I had no idea what condition they were in. I wore headphones, so I couldn't hear what the baby was hearing through the wall. We covered up the side of the screen that said what the baby was seeing. And I didn't even know what condition that baby was in until the study was over, and then I could look at it. So I was really blind to what condition that baby was in. And we did that a number of different ways because I had a lot writing on my dissertation. I really want it to go one way or another. And in some of the conditions it did, and in some of the conditions it didn't. But again, by keeping the experimenters or the observers blind or unaware of what the hypothesis is, we can really control for this, and we can create equal behaviors for all participants. Another way we can do this is a double-blind experiment. We do this typically more in studies where we're going to have where we don't want them to even know what condition they're in as well. A lot of times we'll do these in kind of medical studies where both the experimenter is blind to what they're in, and the participants. Nobody knows what condition anybody's in. So the pills look the same. But some of the pills are placebos and some of the pills are actually have the medication, is the best example of a double-blind experiment. And only, someone who is nowhere connected to the actual data collection at all knows who's getting what, so that they're not able to influence in any way any outcome of the study. And then the participant doesn't know either. This allows us to control for a couple of things. It allows us to control for demand characteristics. So the placebo effect is the idea that you think that medicine's going to help. And if I'm getting the medicine, then the medicine's going to help. And so everybody takes a pill and you think you're getting the medicine, so that the placebo effect, whatever effect it has, is equal amongst the two groups. And then the effect of the medication then should be up above that. So those are examples of controlling for demand characteristics. It also controls for experimenter effects, because the experimenter doesn't know what condition that they're in. And so again, we're allowed to control that as well. It also allows us to rule out that the participant or the experimenter's expectancies as these alternative explanations, because they are controlled for and they had no idea what condition they were in or what condition they were administering to a participant. It's no way that it can be an alternative explanation. It can't be just taking a pill matters, because nobody knew what pill they were taking. Everybody took a pill. It can't be that, having somebody think that you're going to get well, it's going to make you get well. Those kinds of things. So these double-blind experiments allow us to control for both of those things and allows us to eliminate a lot of alternative explanations. Communication between two or more groups, can be a problem as well and can really threaten internal validity. This occurs when there is communication between these groups. So if you are giving your treatment group, you're doing a study about some sort of parental training class, and you have a group of parents, it's your treatment group, and they meet once a week and they go over parenting techniques, really successful parenting techniques, and they do role playing and all these kinds of things in your treatment group. And you have another group of parents that also meet once a week, but they just sit around and talk about parenting. They're not really getting any kind of training. It's more just a kind of a talking, because talking to other parents is helpful. And so they're getting that same kind of experience that the treatment group is getting, but they're not getting the additional actual treatment of the parental skills and those sorts of things. But say your parenting treatment group starts talking to your parenting control group. My kid did that the other day, and you know what I did? What I learned in class. I did this parenting skill that I learned in my class, and it really, really worked. And they tell, the person from parenting control group about it. And, now the person in parenting control group knows that wonderful new parenting technique. And so they may be doing that as well. So we create a situation where there's communication between these two groups. And we have three possible outcomes for that. We can have resentment. Well, why do you get the parenting classes? Why are you getting these parenting benefits? And I get to sit here and talk to a bunch of idiots who don't know what they're talking about. Again, this is the idea of resentment. I don't really think that people are doing this. There can be rivalry. You know what? They're getting the treatment group, but you know what? I am just as good a parent as they are, and so I'm going to out-parent them. And they try to beat the other person because they know that they're in the control condition and the other's in the treatment condition. It causes differential behavior. Or we can have diffusion, which is the example of, here's this parenting technique I learned, where we're spreading this treatment effects to the control condition. All three of these outcomes create differential patterns of behavior that are then become alternative explanations for differences between our groups. We've muddied the waters. There's more noise. It's harder to see our signal. And because of that, that's why communication between groups is a threat to internal validity. So those are our main threats to internal validity. Again, I hope you've reviewed the online tutorial as well on that. And please let me know if you have any questions that you would like to address. So now we know about between subjects designs. We've got these two groups. We've done our study. We have this data. Well, now what? Now what do we do? Obviously, the other part of this class is statistics. So we won't actually do the statistics here, but we'll start talking about it. What we typically do is we look at the two groups' mean differences. We look at group A versus group B, and we look at their means. Are they different? Are they different enough? Is how we typically do this. So we'll calculate a mean for each group, and the easiest way to do this then is a t-test. And again, we'll get into this in the subsequent chapters. Two groups really allow you to maximize the difference between the two because you can choose opposite extremes. You can choose no treatment, high treatment. You can create these really maximized two groups. They're very, very simple and they're very, very helpful, but they're not detailed. Because they're not detailed, you may not be getting the whole picture. So while you might be choosing the highest dose and no dose, that's just telling you the difference between the highest dose and no dose. It doesn't tell you anything in between. This graph from your book kind of shows you the issues of what's in between. So if you did your study with these two opposites, if you did it with the high dose of 4 and the low dose of 0, then we're looking at those differences of A and E on the graph. And there's a difference there, and we could maybe it's even a statistically significant difference. And we can say, okay, our medication worked, it helped. We saw a increase in whatever in this activity level that we were looking for. This was helpful. But because we only had two groups, we don't know that E is not actually our most optimal dose. If we're trying to get the highest activity level, then C is actually, dose 2 is actually our most beneficial dose. So in this example, if we had done maybe a 3-group design where we did 0, 2, and 4, we could see that. If we had done a 5-group design where we have 0, 1, 2, 3, and 4, we can get a better picture of what this looks like. So it really is creating a situation where you're trying to balance between the two. You want to create a situation where you can really see these differences and see them nicely, and these opposites can usually allow you to do this. But you don't get a detailed picture, and you don't get to see the nuances in it. If you do a study with all 5 of these, you get to see the nuances in it. But they also start to cancel each other out. They're not that different. C and D are hardly different at all. So again, we lose some of that, and statistically, that's going to make it harder to see a difference between these. So in this study, kind of maybe the best option would really be ACE, where we're getting a little bit more detailed group here, but we're not really muddying the waters with B and D as well. B and D aren't telling us as much new as adding that 3rd group, C, did. So again, this is hypothetical data that this is from your textbook, but it just is talking about that nuance, that issue between having 2 groups or having too many groups, and where you get in there and what is most appropriate. And again, that's going to be based on the individual research question. When you're comparing means for more than 2 groups, we have what we call a single factor multiple group design. That means the factor and it's important to know this now, factor just means independent variable. So we have one independent variable that has multiple groups. We have multiple levels of one independent variable. So from our drug dose picture from before, we're looking at A, C, and E. We've got all 3 of those groups. We have one independent variable, which is drug dosage, and we have 3 different levels of drug dosage. We then do just what we did with the two groups, and we calculate a mean for each group, and we'll compare those groups. But how we compare them is different. We can no longer compare them with a t-test. T-test can only be done for two groups. Now what we're going to compare them with is an ANOVA. ANOVA stands for Analysis of Variance. And you can see I've underlined where the letters come from in ANOVA, so you can really understand that. And so what we're really doing is we're looking at the variance and looking at the difference between the variance within each of our groups and the variance between our groups. Because we have more levels, it gives us stronger evidence for cause and effect. It gives us again, more independent, more internal validity. But again, you can really water this down. If you had done, A through E, if you had done all 5 of those levels, you really have watered down your data quite a bit. And a lot of those differences can be lost then. So you can have too many groups. It really is creating the struggle is, with research as a whole, is balancing. It's a balancing act. And this is just another situation that we're trying to balance the number of levels of our independent variable to get a really good understanding of our data, to get a really good understanding of the phenomena, but not water it down with too many levels. So I started to draw this on the board one of the first days in class, and this is just part of the table I drew. But this is kind of how it breaks down. So if you're doing a between subjects design and you have 2 conditions, or you have 2 levels of your independent variable, you can do a t-test for independent groups. That's the statistic that's best used. If you have 2 or more conditions, you're going to use ANOVA. So you can use ANOVA with 2 groups. If you have a treatment condition and a control condition, you can use an ANOVA. You can do an ANOVA on that, but it really is a lot more complicated, so a t-test is just as easy. But if you have 3 groups, you have to do an ANOVA. You have lost that option for a t-test. And when we get into those statistics, you'll be able to see why a little bit better. Now note, this is only for numerical data. This is only for things that I can calculate an actual mean for. If I can't calculate a mean for it, I'm not going to be able to use either a t-test or an ANOVA, because both of those are essentially looking at the differences between means. So if I don't have a numerical score, if I can't do a T or an F test, if I'm not using numerical data, say if I'm using nominal data, such as gender or academic major or occupation, any of those things, that's nominal data. I can't calculate a mean of male and female then. Because what is that? You don't have any numbers. You have counts, but you don't have any numbers. Academic major, I can't calculate a mean of psychology versus chemistry. Those aren't things that I can calculate. The same goes for ordinal data. College class, are you a freshman, a sophomore, a junior, a senior? That's not something I can calculate a mean for either. Birth order, are you the first child, the second child? Are you a third child? Those sorts of things. Those aren't things that you can calculate means for. Performance is another one, low, medium, high. In these situations, you're not going to be able to use a t-test or an f-test or a ANOVA. You're going to need to use a different statistical method. A lot of times they'll use chi-squared as a goodness of fit test. It really is a nice statistical measure that you can use to kind of address these kinds of issues. So it is important to remember that when you're doing any kind of experimental design, if you don't have numerical data, neither a t-test nor an ANOVA is going to work. Here's another example from your text where they really are dealing with this non-numerical data. This is research from an experiment. We're still dealing with an experiment. We just don't have numerical data. Where they asked 3 questions of witnesses about the speeds of cars they observed in a collision. The dependent variable was the response to the question. So you got yes or no. Well, I can't calculate a mean on yes, and I can't calculate a mean on no. I get the number of people that said yes and the number of people that said no. And then what I can do is use the chi-squared goodness of fit test, remember, to compare the control condition, the hit condition, to the smashed into condition, and see if there's a difference then between those 3 based on their yes-no responses. So again, you cannot do a t-test or an ANOVA with non-numerical data. You're going to need to use a different statistical method. I hope this was helpful. Please let me know if you have any questions, and we can discuss them either on the discussion board on D2L, via email, or in my office hours. I will see you Wednesday.